Thank you. Okay, so
lights. So lights are okay, right?
Hmm?
Lights are okay.
Yes. Everyone see? Okay. My name is Terry Johnson. I have a chemical engineering degree. I used to work as an environmental engineer, and I now teach in a bioengineering department, so I'm kind of a dilettante. My original work in bioengineering was in liver tissue engineering, but I've been doing a lot lately in synthetic biology. And what synthetic biology, why is it of interest to an engineer? Well, in each of you, there's about 10 trillion cells, but those are human cells, and 100 trillion microorganisms, mostly in your gut, the place you don't want to think about if you've localized it correctly. About 1,000 different species in each of your guts, and the balance of species has health effects, actually. So you're more other kinds of cells than you are your own cells. And I think the computer has done something weird. Okay. To give you an idea of some of the things that microorganisms can survive, this is a temperature line. They can survive from -17 to 266 degrees Fahrenheit. That's snow, that's boiling water. They can survive pHs from -0.6 to 11.5. That's stomach acid, that's milk of magnesia. And they can survive pressures of 0 to 30,000 psi. That is space, and that is the bottom of the deepest part of the ocean. So there's incredible resiliency in different kinds of microorganisms. So we are interested in microorganisms because of that. Many of you have heard, by the way, I'm not saying you want to build hyper-intelligent monkeys. I am saying that if you give a monkey a typewriter and you have a million or bajillion or quintillion or infinite number of monkeys and you give them an infinite amount of time, the joke goes, you will recreate the collected works of Shakespeare. This has actually happened on this planet. There's the oceans alone. 10 to the 30, 10 followed by 30 zeros, microorganisms in the ocean. It's probably approaching the amount of water molecules that are in this room, including the water molecules in your bodies. It's a tremendous amount of life. And they've had 4 times 10 to the 9 years to futz around. They've created a lot of interesting solutions to difficult problems. Thus, the interest of an engineer. Engineers like to solve problems. There are a lot of solutions to problems out there in these microorganisms. Where do we find these things? All over the place. The J. Craig Venter Institute is actually going through the ocean trying to pick up interesting microorganisms. These are sailing routes that they've chosen to look for interesting microorganisms. We look for places that are odd, like hydrothermal vents or the Iron Mountain mine, where microorganisms are surviving under weird circumstances. They may have developed interesting ways to survive in those circumstances. We can also look at other forms of life like plants. This is woad, which we're going to talk about a little bit later. And I haven't even talked about mammalian life, like penguins. And I use that because I like penguins. But there's a lot of different kinds of life on the planet that those different kinds of life have come up with different ways to solve problems associated with surviving. Life that was bad at surviving is no longer here, right? So beating the cell at its own game. Cells are very good at what they do. They've had 4 billion years and many, many cycles, many iterations of improvement to figure out how to do things. But they don't always do what we want them to do. Anybody that has had a bacterial infection is very clear on this. Oftentimes, they don't do what we want them to do. They do what works for them. So can we build new cells? Can we build cells that do what we need them to do to solve problems? Genetic selection is how this used to be done, right? How do you come up with life that does interesting things? Well, you wait for it to randomly appear. Genetic selection, you could say, this is a 10,000-year-old cave painting. You can see the hunting dogs near the stag. There are suggestions that dogs have co-evolved along with us, that we've been affecting canine evolution for a very long time. About 10,000 years of corn, that's what corn used to look like on your left. And you're all, I think, familiar with corn now. That is because people have been selecting corn for size and deliciousness for 10,000 years. And about 50,000 years of dating. Now we have genetic engineering, which is a different way. Instead of waiting for random evolution, basically random mutations to occur that happen to be good. We try to build things that are good. In 1973, antibiotic resistance in E. coli was transferred. Very, very kind of goofy technique. You just busted the heck out of DNA and you dumped it into E. coli of a certain kind that was capable of taking up that DNA, and some of them were able to use that DNA to survive. In 1978, we were producing human insulin in E. coli. Actually, the technique was a little more complicated than that. We were building precursors for human insulin and then doing some reactions outside. That's Genentech right there. None of those people have to work. Today, we produce over 6 tons of insulin every year via biotechnology. And if we didn't do that, we'd have to get it from animals. We have to get it from animals. Factory farming would be a lot worse than it is today, and it's pretty bad. So, how do we do this genetic engineering? Do we have a cell? A cell has DNA. DNA gets transcribed into RNA. RNA gets translated into a protein. The way to think about this, if I had a computer, I have an installed program. It's sitting on a hard drive somewhere. I open that program, and I have a program window, an instance of the program that's live and in memory. And then I get an output to the screen from that program window, which might be an adorable puppy GIF. But that's how I get something that I want. So let's say what I want is some kind of particular protein. I'm going to have to put DNA into the cell so that that stuff will get made. It would look something like this. I want to take a protein from jellyfish, red fluorescent protein. If you shine it with black light, it will glow red, right? And I want to make bacteria that make that. I'm going to install the RFP DNA, which is going to make RFP RNA, which is going to make the actual RFP protein, which is what I see. So I'm just basically putting instructions in for something that the cell doesn't usually make. And you get something like this. These are bacteria, bacterial colonies actually, that glow red under a black light. Okay, this is from iGEM team 2010 in Alberta. So we're going to talk a little bit about DNA. This is a nice fancy GIF of DNA kind of circling. DNA is actually made of bases, adenine, cytosine, guanine, and thymine. Some of you may remember that. But in reality, I want to note that when we think of DNA, typically we just think of it as strings of letters. It's actually a molecule. It's got shape, it's got charge, it's got all of this stuff. We already abstract DNA. When we talk about DNA, it's just strings of letters. But those strings of letters are representations for an actual physical thing in space. Small physical thing, but a physical thing in space. So keep that in mind as we go on. We're already abstracting what's going on. So this is part of the DNA for making bacteria that glow red, not the whole thing. Yeah. That doesn't look very fun to design. I can make it a little bit easier by color coding parts of it. The blue part is a promoter. It is associated with sort of getting something started to make it. The green part is a terminator, which tells the cell machinery we're done here. And the red part actually says this is what red fluorescent protein should be made of. That's the instructions for making it. So I've got a cell, and I put in a little piece of DNA, that little round bit, to make red fluorescent protein, and ta-da, it makes the red fluorescent protein. The instructions for that protein are now there. But let's say, for example, I want to make another cell that also makes red fluorescent protein, but makes a lot of it. It's very, very bright red. Or I want to make a cell that doesn't do anything, unless it senses a pollutant. And only under those circumstances does it turn red. Different function requires new DNA. New DNA requires more design work. I have to start all over, and I have to think in terms of that. There's a giant long string of letters. How am I going to edit this string of letters? This is not fun. So what I'm going to suggest is really how we should be thinking about DNA to solve this problem. That's the question that synthetic biologists often ask. So what if I want to build something really complicated? This is artemisinin. It is a relatively complicated molecule. It's on the small side, but it's got some issues with it. And I want to make artemisinin for a number of reasons, in particular because of malaria. It is a treatment for many forms of malaria, which, by the way, the mosquito, the Gates Foundation recently showed data that it is the most dangerous animal on Earth. right? So the next time you go like that, you are hunting the most dangerous animal on Earth. So how hard could it be? I want to make this molecule, right? That. This is not a complete picture of all the things that have to happen inside of a cell to make artemisinin. It is a partial picture of all of those things. A lot of those are from different species entirely. Some of those are genes from plants, some of those are genes from other yeasts, some of those are genes from elsewhere in the kingdom of life on the planet. I would have to install all of this stuff into the yeast, and then I need to tune the yeast, because once you've installed all of this stuff, you have this finely tuned machine, and then you dumped a bunch of other stuff into it and said, make this too. So you very often have to do a little bit of retuning of the engine, so to speak, to get it to function the way you want to. And as you can imagine, this process is something like this. Designing that as a bunch of letters is horrifying. So who can teach us a better way? How should we be thinking about DNA to solve problems like this? Why? Okay, building a Bjorken. I chose Bjorken for 2 reasons. 1, I like saying Bjorken. 2, bioengineering, it is a medicine cabinet. That's a Bjorken. How many of you have made anything from IKEA? So you're familiar with this little dude? Yeah. And the parts lists. So a question that I have a device that I want to build, and I have parts which are what I want to build it with. There. I'm going to take one of those parts. What's left out of those instructions? Well, you take a look at that screw. Doesn't say the length of the screw, it doesn't say the number of turns, it doesn't say the angle of the turn, it doesn't say that it's made out of this standard stainless steel, it doesn't say that it has a breaking strength of this plus or minus this. It doesn't say any of those things because none of you care, right? It only says there's this many parts and this part goes in here. Why? Because you don't make screws. You put together the Bjorken. You don't make the screws, you use them. So parts. The maker of a part needs to know how the part was built. The user of a part needs to know how the part interacts with other parts. And this is a fundamental difference about looking at DNA. But what does it give you? So I've got DNA parts. Instead of that, I have a promoter part attached to a protein part, which is RFP, attached to a terminator part. And the advantage that this gives me is that I have this. I could put in a promoter to make a little bit of RFP. I could make a promoter to make lots of RFP. I just swap that in. Or another promoter to make RFP in response to heat, or a 4th promoter to make it in response to a pollutant. On the other hand, I could use the exact same structure and make green cells by swapping in a different protein, or yellow cells by swapping in a different protein. So it's more about the combination of the parts and how they interact. I don't have to constantly be looking at them as long strings of letters, which in and of itself is an abstraction because in reality it's a giant molecule that in us is all wound up and we just think of it in terms of letters. The second level of abstraction that synthetic biologists use is we should look at those long strings of letters as parts based on their functions and how they interact. So things that synthetic biology isn't, or my personal favorite, in general, synthetic biologists work typically with yeast and bacteria. And they're working with yeast and bacteria to produce things or to be better at producing things that they already produce. Sometimes we do work with mammalian cells, and usually that's for healthcare reasons, right? Like for example, if we were able to pull an immune cell out of the body, reprogram it so that we put it back in you and it hit the cancer, that would be the sorts of things that synthetic biologists would do with mammalian cells. Synthetic biologists just don't work with actual creatures. We work with individual cells. So to give you an idea, I've worked with teams of undergraduates at UC Berkeley. I'm going to start with a project we did in my first year in 2008, and then with a project that we did last year. So these are teams of undergraduates working with me and several other faculty at UC Berkeley in the bioengineering department. That's lambda-phage. It is a virus, and you could drink it because it won't do anything to you. It's a virus that only attacks bacteria. And if you take lambdaphage and you add bacteria to it, the bacteria that it recognizes, it will basically hijack that bacteria and force it to make a bunch of phage. You can see it's produced a bunch of phage inside of it. But the phage needs to get outside to complete its life cycle, right? And it does that by breaking open the bacteria like a piñata so that the phages can escape and infect other bacteria. Now we're interested in this because what if we, for a number of reasons, but what if we make a bacteria that's got a drug inside it? We've basically made the cell make a drug. We now need to get the drug outside of the cell. There's different ways we can do that. We can use chemicals, but those are expensive and they sometimes damage the drug, right? They're also sometimes environmentally not so great. But we could take bacteria that makes drug and add phage genes to it, so when we want it to, it will break itself apart. This is a self-lysis device, a self-break-apart device. When it's on, it becomes clear because the cells have broken apart, releasing their contents. When it's off, the cells are healthy. It's cloudy. There's lots of cells in there. So we can effectively say cells, when you have made what we want you to make, break apart so that we can get at what is inside of you without any sort of environmental problems. How many people here are wearing blue jeans tonight? I'm not. I should raise my hand. All right. Blue jeans are dyed with oil. Actually, it's indigo, but indigo is a petroleum product. It used to come from the plant woad, but that is too expensive. So typically you take petroleum, goes through a bunch of steps that are very high energy and use a bunch of stuff that I can't pronounce readily. But once you go through all of those processes, you get indigo. Indigo is blue and insoluble, so to get it into your genes, you make into gluco-indigo. You reduce it. So you add a harsh reducing agent, which ends up in rivers in China, and effectively it turns into a white indigo, which dissolves in water. You expose denim to that, and then you leave the denim out to dry. And as it dries, it oxidizes, back into the actual colored indigo. That's how genes are made now. Last year, our group of students took cotton cloth, that's our little bear cloth, and what they did was they made something called indican, and they showed that they could make indican in yeast. And indican is like indigo, but it happens to be a soluble form, and it's one that you can make a decent amount of. And they also made an enzyme in separate cells called beta-glucosidase. Beta-glucosidase takes that indican and cuts it in such a way that it becomes something that will oxidize directly into indigo. So effectively, instead of having to make it soluble, you make a soluble biological form, and then you are able to unleash it by adding an enzyme that brings it back to something that will turn blue. And the idea would be that if you were to expose to the indican and then expose to the beta-glucosidase, you would get blue genes. And that's what happened. This is, by the way, over probably 60 or 70 minutes. This is a sped up video. Actually 30 for this one, I apologize. They were able to take these, wash them. It was a permanent dyeing. You could wash it in soap, you could wash it in water, you could wash it in ethanol, and they would stay dyed. And there's at least one blue jeans company that is interested in this process. So, what's next? Okay, so we've talked about this sort of new abstraction. If you're going to build an engine, you're going to model it in something like SolidWorks. You're going to build it in a computer. If you build a chemical plant or a fermentation plant, you're going to actually construct it first in a program called Aspen or something similar. And if you're going to make a circuit, you're going to model that circuit first in something like HSpice. These are just specialized programs for make a circuit, make a chemical plant, make an engine. Now, if we're making larger and larger constructs that are more and more complex in synthetic biology, we would want to take advantage of something like this. Because if you think about it, all of these systems basically say, how do these parts work together with one another? How does a piston work with the rest of the engine? How does a heat exchanger work with a chemical reactor? How does a capacitor work with a resistor? Basically, how do the parts interact? And that is the abstraction that we've been talking about. We can also use this to look for combinations that might present subtle safety risks. So we basically want to give tools that allow people to build more quickly, better and more safely. And one of the initiatives that's at Berkeley is to make a program called Clotho that does exactly that. It warns you when you're using genes that have been associated with problems in other organisms. It basically helps to give you an idea of what sorts of products that you can expect, especially in combination with other programs. And it's a design tool for being able to build better and better systems for solving problems. So the goal in all of this, the reason why we're interested in biology as engineers, is to use cells as solutions for problems. We've been doing this for quite some time. The very first technology was brewing beer, depending on who you ask. It's a technology that's been going around for thousands of years. So we're very good at it. We'd like to take advantage of it to do things other than merely getting drunk. And there are a number of chemicals, both commodity and specialty, that are made using techniques, biological techniques like synthetic biology. We would like in the next 10 to 20 years to expand that significantly. Thank you for coming. If you have any questions, I'd be happy to take them. Yes?
Can you say a few words about your book?
So I was a co-author on that book. My friend Kyle, who manages a translational medicine master's degree at UC Berkeley, he was in his PhD program at the time, came to me with the title, and he said, we should write this because I've got a really good title. And I said, that is a good title. That's the reason why his name comes first on the book, is because he titled it. It's basically an exploration of biotechnology in video games and movies and television, and we'll sort of give examples from popular culture and talk about the science that we think inspired them. Sometimes we'll say when they're way off, and sometimes we'll point out when they really did an excellent job of sort of extrapolating from the facts. Available on Amazon.
Yes, question.
What's phage? One of the terms you used was pH.
Oh, phage.
Phage, sorry.
Phage is a virus, but it's a virus that targets bacteria. Yeah. And there are, underneath the world that we live in, there is a tremendous diversity of bacteria and a tremendous diversity of phage trying to take advantage of the fact that there's all those bacteria. And phage are sometimes used just as tools in biotechnology because you can use them to manipulate bacteria. But if you were to quantify the number of phage in this room, I'm guessing it would be significant.
Can you just clarify the distinction between a molecular biologist and a synthetic biologist?
No, I can't. So molecular biology, there was a paper that came out a couple of weeks ago where a molecular biologist was making fun of the lingo that synthetic biology has come up with, that there's already lingo in molecular biology for. But anytime there's, especially in academia, but anytime there's a new kid on the block, like we're not going to call ourselves molecular biologists. You're the old fuddies. We're going to call ourselves synthetic biologists. We're going to invent our own language because then you have to be in the club to understand what we're talking about. Also, sometimes because we're using fundamentally different viewpoints to look at things and construct things. So I don't think there is a hard and fast rule. I would say instead, depending on who you talk to and what conferences you go to and what techniques maybe you use, you will either self-identify as a molecular biologist or a synthetic biologist. I will work with anybody who can do stuff that I need.
From your talk, it seems like it's about end product in this way, like what your end goal is, like you're going to manufacture. I can't quite tell. I'm always confused about this.
Well, that
suddenly becomes synthetic biology, but it seems so.
Yeah, that might be an engineering bias too, because I tend to frame things as building things as solutions for problems, but there are also synthetic biologists that are based mostly on building tools to manipulate cells on a sort of higher level or with a better degree of control. So I guess the answer to that question is I don't know.
Yeah.
And it's one of those, somebody will write a PhD in culture of science.
Yeah, they will.
And it will be based on this. It'll happen in about 5 years.
Okay, thank you very much.
Thank you.